Welcome, curious minds, to another deep dive. If you just pause for a second, look around you, what do you see? Maybe a tree, a pet, a houseplant. On the surface, a majestic tiger, swaying seaweed, a fluttering butterfly, a towering oak tree. They all seem incredibly different. Yet our ancient ancestors, long before cells or DNA were even concepts, still sensed something profound connecting them. They called it life, and they've been marveling at its mystery ever since. So today, we're going to peel back those layers of apparent diversity. Our mission on this deep dive is to uncover the astonishing commonalities that lie beneath it all and what those universals tell us about life's fundamental nature and origins. We're drawing our insights from an extensive and incredibly important source, a key chapter from the Molecular Biology of the Cell, 7th edition.
That's right. And it's a truly fascinating journey we're about to embark on. We'll explore the universal features that define all life on Earth, trace the evolutionary tree of life through the revolutionary lens of genomes, understand the unique complexities of eukaryotic cells, and discover how studying specific model organisms has unlocked our deepest biological secrets. As our source beautifully puts it, it's about balancing astonishing variety in individual particulars and astonishing constancy in fundamental mechanisms. It's this paradox that makes biology so endlessly compelling.
So let's just jump right in. At its most fundamental level, what actually defines life? How do you even begin to categorize something as a living organism?
Well, at its core, our source describes organisms as intricately organized chemical factories that possess this extraordinary ability to self-reproduce. And the fundamental self-copying unit of all life on Earth, from the tiniest bacterium to, you and me, is the cell. It's the building block, the tiny self-contained factory that makes everything else possible.
And what's truly mind-boggling about these cellular factories is this universal genetic language they all speak. Every form of life on Earth stores its hereditary information in double-strand DNA, and they all use the exact same 4 nucleotides: adenine, thymine, cytosine, and guanine A, T, C, G. What does that tell us about life's origins?
It's one of the strongest pieces of evidence we have for a universal common ancestral cell. Imagine a single progenitor that existed somewhere between maybe 3.5 and 3.8 billion years ago. The fact that your human DNA can be successfully read and interpreted by bacterial machinery, which diverged from our lineage billions of years ago, is just, what's a profound connection. It's like finding a single ancient alphabet that every language in the world still uses. And the totality of this hereditary information, by the way, for any given organism is what we call its genome.
That shared genetic language is truly profound. But how does this incredibly complex instruction set, this DNA, manage to copy itself so perfectly every single time a cell divides? What's the secret to that faithful duplication?
Right, that's where the elegance of DNA's double-strand structure comes in. Each strand serves as a precise template. Think of it like unzipping a zipper. Each half then guides the creation of a perfect new matching half thanks to those AT and CG pairing rules. It's an incredibly simple yet robust mechanism for copying the blueprint.
Simple but powerful.
Exactly, ensuring that the genetic information is passed on almost flawlessly from one generation of cells to the next.
So you've got this perfect blueprint being copied. But having the instructions is one thing. How do those static instructions actually become the dynamic, bustling activity of a living cell? How do they get put to use?
That's the process of gene expression. It starts with transcription, where segments of DNA are used as templates to synthesize RNA molecules. It's like making a temporary working copy. For messenger RNA or mRNA, uracil U replaces thymine. These mRNA transcripts are described as mass-produced and disposable intermediates that then guide protein synthesis. But it's worth noting that not all RNA molecules code for proteins. Some have direct functional roles in the cell, like those that form the structural core of the ribosome itself.
So if DNA holds the master instructions and RNA carries the message, what are the actual doers in the cell? What performs all those countless functions that make life possible?
Those would be the proteins. They're the cell's true workhorses. They're long, unbranched chains made of 20 different amino acids. And here's where it gets truly amazing. The linear sequence of these amino acids dictates a unique three-dimensional shape for each protein.
The shape determines function.
Exactly, and that specific shape, in turn, determines its precise function. We're talking about everything from enzymes that catalyze chemical reactions, to structural components that give cells their shape, to proteins that generate movement, and even those that sense signals from the outside world. It's all down to the protein shape.
It sounds like a beautifully orchestrated system where everything depends on everything else. Is that why our source talks about the autocatalytic nature of life? What does that mean?
Precisely. It's a self-sustaining, self-replicating loop. DNA and RNA provide the sequence information, the instructions. But it's the proteins that catalyze almost all the chemical reactions needed to put those instructions into action. And this includes the reactions that synthesize more DNA, RNA, and proteins themselves.
Wow.
It's a closed-loop system where the products are also the tool for creating more of the products, autocatalytic.
And the genetic code itself, the language that translates mRNA sequences into amino acids. It's read in triplets or codons. Each triplet specifies a single amino acid.
Yeah.
But what's truly astonishing, and a bit odd, is that this universal code is described as strangely arbitrary. It's thought to reflect frozen accidents from early life. What does it mean for something so fundamental to be arbitrary? Does it suggest life could have evolved entirely differently?
Oh, absolutely. Imagine if we decided tomorrow to change the meaning of stop at a traffic light. The chaos would be immense. Life's genetic code made an arbitrary choice billions of years ago. A particular triplet just happened to code for a particular amino acid, and because it worked, evolution essentially locked it in. Any change now would be catastrophic for the cell.
A frozen accident. That's a great way to put it.
It's a profound example of how initial chance events can shape the entire trajectory of life, leading to this deep universality we see today, and the decoding machinery for all this that's handled by specialized transfer RNAs or tRNAs and the incredibly complex ribosomes.
So if we define a gene as a segment of DNA coding for a protein or a functional RNA, how does a cell decide when and where to actually express these genes? It can't just be full throttle all the time, right?
No. Absolutely not. That would be incredibly wasteful and probably harmful. The cell's ability to adjust when and where these are made is crucial. That's gene regulation. Cells don't just churn out everything at full tilt. They precisely control gene expression based on their immediate needs. This is achieved through regulatory DNA sequences.
Like switches.
Sort of, yeah. These sequences bind to specific proteins called transcription regulators, which then fine-tune the rates of transcription for individual genes like a molecular dimmer switch.
Now, all this complex activity, copying DNA, making RNA, building proteins, regulating genes, it requires immense energy. It sounds like a cell is constantly fighting against chaos, pushing back against the universe's tendency toward disorder. How does it manage that incredible feat?
Exactly. You've hit on a key point. Think of it like a meticulous, bustling kitchen that always has to keep ordering ingredients and cooking. Otherwise, it quickly becomes a mess. A living cell is a dynamic chemical system operating far, far from equilibrium, meaning it's constantly expending energy just to maintain its organized state. Without that continual input of free energy, whether it's from sunlight for plants or chemical bonds in food for us, the random thermal motion of molecules will just break everything down. It's a continuous molecular battle against entropy.
So how does it harness that energy?
Well, our source uses a great analogy. Imagine molecules as spring-loaded traps, ready to snap into a more stable, lower energy state when they meet their proper partners. That snapping releases energy, driving the ordered processes of life.
And for all this intricate work, the universal currency of energy, no matter the source, is ATP, adenosine triphosphate. So the cell is truly a sophisticated biochemical factory, manipulating a similar collection of small organic molecules. What about its outer limits? Every factory needs a wall, a boundary, right, to control what comes in and out.
And that's the plasma membrane. It's an essential selective barrier that allows the cell to concentrate nutrients from its surroundings and keep what it needs, while efficiently pushing out waste. Without it, the cell couldn't maintain its integrity. And the remarkable thing is how these membranes form. They're made of amphiphilic molecules like phospholipids, which have a water-soluble head and water-insoluble tails.
Hydrophilic and hydrophobic.
Exactly. And these naturally self-assemble into bilayers in water, forming closed vesicles. It's an amazing act of self-organization, driven purely by the chemistry of water and these molecules.
And how do things get in and out of this selective barrier? Are there just, like, holes?
No, it's far more sophisticated than just holes. All cells have specialized transport proteins embedded in their plasma membrane. These proteins are so well conserved across evolution that we can see their family resemblances even in the most distantly related organisms. They largely determine what enters and leaves the cell, working in concert with the enzymes inside that dictate the chemical reactions.
And you mentioned thermal motion, things just bouncing around randomly.
Right. At the incredibly tiny scale inside a cell, remember, we're talking micrometers. Molecules are never truly still. They're constantly being jostled and bombarded by other molecules, especially water, in this chaotic, random dance called Brownian motion. Imagine a tiny rubber duck floating in a very agitated bathtub. It's constantly bumping into the sides and other toys, seemingly without direction. This constant motion actually drives diffusion and reaction rates as molecules collide.
It's amazing that randomness, Brownian motion, can actually be harnessed for directed movement. This Brownian ratchet sounds almost like magic. How does that work?
It's not magic, but it's brilliant biology. Imagine a crowd pushing randomly against a door. Normally it just wobbles. But if you had a system that could quickly put a tiny wedge in the gap every time the door happened to swing outwards, preventing it from swinging back.
You'd slowly nudge it open?
Exactly. You'd slowly but surely nudge that door open. That's essentially what some cellular machinery, like parts of the cytoskeleton, can do. Exploit those random thermal fluctuations, stabilizing favorable movements and preventing backward slips, leading to directed motion like crawling. It's ingenious.
And to wrap up our look at these universal features, you mentioned the bacterium Mycoplasma genitalium earlier. It has one of the smallest known genomes. What does that minimalist organism teach us about the essence of cell biology?
It's a fantastic example of life boiled down to its bare essentials. This parasite has only 525 genes. That's about one chapter of our textbook's worth of information, basically. It still has to make all its DNA, RNA, and proteins and manage energy, but it shows us that while cell biology is complex, it is not unimaginably so. It's a reminder of life's fundamental elegance and efficiency.
Okay, so those are the fundamental building blocks inside the cell. But zooming out, these tiny factories have had a massive impact on the planet itself, haven't they?
Oh, absolutely. When you take billions of these tiny, intricate factories operating for billions of years across the planet, their impact isn't just internal, it profoundly reshapes the entire Earth. Our oxygen-rich atmosphere, the vast deposits of coal and oil, even geological formations like chalk and limestone, all products, directly or indirectly, of past biological activity. But what's often overlooked, as the source points out, is that most organisms on our planet are microscopic and inhabit extreme environments, often unseen and unculturable. The familiar world of plants and animals is just a tiny fraction of the total diversity.
That's a truly humbling thought. How do we even begin to map this unseen majority? Because for a long time, we classified life based on what we could literally see. But now, with modern DNA sequencing, how has that changed our understanding of the tree of life?
It's been a complete revolution, honestly. Instead of relying on outward appearances, does it have fur, does it have leaves, we can now make objective, quantitative comparisons of DNA sequences to figure out how closely organisms are related.
Like a genetic family tree.
Exactly. It's like discovering a universal genetic blueprint for all species and comparing the subtle variations in their code to determine their lineage. Much more objective.
And for constructing this comprehensive tree of life, you need a molecular ruler, a segment of DNA that is easily recognized in the genomes of all organisms. What's the go-to for that?
That would be ribosomal RNA, or rRNA. It fits the bill perfectly. It's a core component of ribosomes, which are fundamental in all life, and its sequence is highly conserved, meaning it changes very slowly over time.
So it's reliable for deep history.
Extremely reliable. The more similar the rRNA sequences between two organisms, the more recently they diverge from a common ancestor. It's our best molecular clock for tracing deep evolutionary relationships.
And what has this molecular ruler revealed about life's fundamental divisions? We often think of plants and animals as the big categories, but it sounds like there are some surprises in store.
Big surprises indeed. This approach has revealed the three major domains of life: eukaryotes, bacteria, and archaea. It's a huge revelation for most people to realize just how much diversity exists within bacteria and archaea. They represent the vast majority of life, making the familiar world of plants and animals seem like a very small slice of the total biological pie.
So let's briefly describe these three domains. What sets a eukaryote apart from a prokaryote? That's the main split, right? Well, two of them are prokaryotes.
Right, so eukaryotes, from the Greek truly nucleated, are defined by having their DNA enclosed in a nucleus. They're typically much larger than bacteria and archaea and contain various other membrane-bound organelles, those little compartments we talked about that give them internal complexity. Prokaryotes, which includes both bacteria and archaea, lack a nucleus and most of these internal compartments. Their DNA just floats in the cytoplasm.
Okay, let's take a closer look at bacteria first. We often only hear about them when they make us sick, but they're incredibly diverse and crucial, right?
Absolutely. They're usually tiny, often spherical or rod-shaped, measuring just a few micrometers, and typically have a tough cell wall for protection. They can live as independent individuals and don't let their size fool you. They are astonishingly diverse in their biochemical capabilities. Some can do photosynthesis. Others feed on simple inorganic nutrients like hydrogen gas or iron ions, literally eating rock. They have a massive impact on human health, both good and bad.
like our gut microbiome.
Exactly. And they are increasingly used as factories in genetic engineering, creating everything from medicines to biofuels. Think of Escherichia coli, or E. coli, a common inhabitant of our gut, or Anabaena cylindrica, a photosynthetic bacterium that fixes nitrogen from the air, or even Vegetoa, a fascinating bacterium that oxidizes hydrogen sulfide in sulfur springs. Huge variety.
And then there are archaea, which you described as the most mysterious domain. What makes them so enigmatic?
Well, outwardly, they often resemble bacteria in size and shape, but genetically, they're actually much closer to us eukaryotes. It's quite surprising. In fact, some scientists have even proposed only two main domains, bacteria and then a combined archaea-eukaryote domain, reflecting this deep genetic kinship.
Really? So maybe not three domains after all.
It's an ongoing discussion based on how you weigh the genetic evidence, but traditionally it's three. And while initially thought to only inhabit extreme environments like hot springs or highly salty lakes, they were called extremophiles, we now know they are incredibly abundant in common places like soil and seawater, playing major unseen roles in nutrient recycling throughout ecosystems.
The numbers on Earth's biomass distribution really put this in perspective. You mentioned plants account for about 80% of total biomass, animals a mere 0.4%, and humans. Well, we're already 10 times more massive than all wild animals combined, and our numbers are still increasing.
Staggering.
It really underscores the incredible interdependence of all life forms. We're a tiny but very impactful slice.
And this interdependence is also profoundly evident in how organisms obtain energy. You have organotrophic organisms, like animals and fungi, that feed on other living things or their organic products, basically eating carbon that's already been fixed by something else. Then phototrophic organisms, like plants, algae, and some bacteria, harvest sunlight directly.
And the rock eaters.
Right, the lithotrophic organisms, literally feeding on rock, which capture energy from inorganic chemicals. These are often found in extreme anaerobic environments, like the hot hydrothermal vents on the ocean floor. They form entire ecosystems powered by geochemistry, not light, in a world that feels truly alien to us. No sunlight needed down there.
And it's not just energy. Cells rely on each other for matter, too. Consider crucial processes like nitrogen and carbon fixation. Atmospheric N2 and CO2 are incredibly unreactive, requiring significant energy input to make them available for biological molecules. Many cells can't do this, so they depend on others. We depend on plants for carbon and nitrogen, ultimately.
And plants, in turn, depend on nitrogen-fixing bacteria in the soil or in root nodules. This leads to fascinating symbiotic associations, some of which are so close that the partners have lost their separate identities, forming a single composite cell, what we call endosymbiosis, which we'll get to.
This reliance is everywhere.
Absolutely. Which brings us to how genomes diversify over evolutionary time, building this incredible tree of life. At the most basic level, random mutations and errors occur during DNA replication, altering the nucleotide sequence. Most are neutral or harmful, but occasionally a change is beneficial, giving an organism an increased likelihood of survival and reproduction. This cycle of mutation and natural selection is the engine of evolution, constantly driving new ways to exploit the environment.
And the slow-changing genes are key for mapping.
Exactly. It's also why highly conserved genes like rRNA are so crucial for tracing distant relationships, because they change very slowly, indicating their fundamental importance. They're like ancient landmarks in the genome.
So the raw material of evolution is the DNA sequence that already exists. But how exactly does this existing DNA sequence get innovated upon? What are the key mechanisms for creating new genes or new functions?
Our source highlights four main mechanisms of genetic innovation. First, intragenic mutation. Just random changes within an existing gene's DNA sequence, often a single nucleotide swap, insertion, or deletion. Second, gene duplication. An existing gene is accidentally copied, maybe during DNA replication. This is huge because it allows the two copies to diverge in function over time. One can keep the original job, the other is free to experiment, potentially gaining a new function.
So you get spares.
Pretty much, yeah. Spares that can evolve. Third, DNA segment shuffling. Two or more existing genes break and rejoin to form a new hybrid gene with combined functions, like sticking parts of different tools together. And fourth, horizontal DNA transfer. A piece of DNA is transferred between different species, as opposed to the usual vertical transfer from parent to progeny.
Passing genes sideways.
Exactly. This last one is a massive driver of rapid evolution, especially in bacteria. Think antibiotic resistance spreading between different bacterial species. That's often horizontal transfer.
That concept of gene duplication allowing divergence is fascinating. Are there specific terms for these related genes?
Yes, when we compare genes between species, we use specific terms. Orthologs are related genes in different species that derive from a common ancestral gene, likely retaining similar functions. So, your human hemoglobin gene is an ortholog of a mouse hemoglobin gene. Paralogs are duplicated genes within a single genome that have diverged in function. For example, our alpha and beta globin genes, which both make up hemoglobin, are paralogs because they arose from a duplication event in our ancestors and now have slightly different specialized roles.
And homologs.
Homologs is the general term covering both orthologs and paralogs, any genes related by descent. This knowledge is incredibly useful because if we find a new gene, we can often deduce its function by comparing it to a known homologous gene in a well-studied model organism. It's like recognizing a familiar component in a new machine.
And here's a truly remarkable commonality, linking back to our initial theme. Over 200 gene families are common to all three domains of life: eukaryotes, bacteria, and archaea. What does that commonality tell us?
It's incredibly powerful evidence. It strongly suggests a shared inheritance from that last universal common ancestor, or LECA. These aren't just random genes, they are the core machinery genes, fundamental to DNA replication, protein synthesis, energy metabolism, the absolute essentials for life. It paints a vivid picture of a single ancestral cell whose basic operating system has been passed down, evolving and diversifying for billions of years.
Amazing continuity.
Truly. Now let's shift our focus specifically to the eukaryotic cell, which, as we briefly mentioned, is generally larger and much more structurally complex than bacterial and archaeal cells, boasting numerous intracellular organelles. It's like comparing a simple tent to a multi-room house with specialized workshops.
So inside these incredible eukaryotic cells, there's a whole city of tiny compartments, each with a specialized job. How do they all work together? Can you take us on a journey, perhaps tracing how a protein might move through this internal factory?
Absolutely. Let's try. At the heart of it all is the nucleus, usually the most conspicuous organelle, which contains most of the cell's DNA packaged into chromosomes. It's surrounded by a double-layered nuclear envelope, perforated by nuclear pores that regulate traffic in and out, essentially the cell's central command center.
Guarding the blueprint.
Precisely. Outside the nucleus, there's the elaborate cytoskeleton, a dynamic network of protein filaments, actin filaments, microtubules, and intermediate filaments. This provides mechanical strength, drives cell movement, and is crucial for cell division. Unlike prokaryotes, eukaryotic cells can change shape rapidly, and some can even engulf other cells through a process called phagocytosis, like an amoeba eating.
In the internal membrane network.
Right. Then you have the network of membrane-enclosed organelles. The endoplasmic reticulum, or ER, is like the initial assembly line where many proteins destined for secretion or for embedding in membranes are first built or start their journey. From there, they often move to the Golgi apparatus, which acts like a sophisticated sorting and packaging center, adding the final touches and directing where each protein needs to go, whether it's to another internal compartment or perhaps shipped out entirely from the cell in a vesicle.
The post office of the cell.
Yeah, something like that. Lysosomes are the cell's recycling plants, small organelles for intracellular digestion, breaking down waste materials and cellular debris. And peroxisomes use hydrogen peroxide to inactivate toxins safely. Materials are continually exchanged between these organelles and with the outside of the cell via small transport vesicles through processes like endocytosis, bringing things in, and exocytosis, sending things out. It's a constant flow of traffic.
Beyond the nucleus and this elaborate transport system, you also mentioned two other key eukaryotic organelles enclosed in double membranes: mitochondria and chloroplasts. What's their unique story?
These are the powerhouses. Mitochondria produce most of the cell's ATP by oxidizing food molecules, essentially acting as the cell's energy generators using oxygen. And in plants and algae, you also find chloroplasts, which perform photosynthesis using sunlight to synthesize carbohydrates. What's truly striking is that in many eukaryotic cells, these two organelles together can take up roughly half the cytoplasm.
Half the volume, just power plants.
Pretty much, yeah. The surrounding fluid is called the cytosol, where ribosomes translate RNA into proteins and many other metabolic reactions occur. But mitochondria and chloroplasts are major players.
This brings us to one of the most compelling stories in biology, the endosymbiotic theory. It says that mitochondria and chloroplasts were once free-living organisms that were swallowed by another cell. How did that happen, and what's the evidence?
It's an incredible tale of ancient cellular cooperation, one of the cornerstones of modern cell biology. Mitochondria are believed to have evolved from an aerobic bacterium, one that could use oxygen efficiently that was captured by an ancient anaerobic cell, likely an archaeon. What's fascinating is the recent culturing of the Asgard archaeon, which has long branching protrusions, providing a glimpse of how such an engulfment might have occurred billions of years ago. It's like one cell literally ate another, but instead of digesting it, they formed a permanent, mutually beneficial partnership. The host provided protection and nutrients. The bacterium provided much more ATP.
A merger, essentially.
A very successful merger. And chloroplasts similarly evolved from a symbiotic photosynthetic bacterium that was engulfed by an ancient eukaryotic cell that already possessed mitochondria.
So plant cells essentially became farmers, no longer needing to hunt for food, unlike many hunting eukaryotic cells which rely solely on mitochondria.
Exactly. This shared history means eukaryotes have hybrid genomes. Most of the DNA is in the nucleus, but small amounts remain in mitochondria and chloroplasts, clearly reflecting their bacterial origins. They still have their own ribosomes and some DNA. In fact, many genes that were originally in the endosymbiont genomes have migrated over billions of years to the host cell's nucleus, but their DNA sequences still show clear evidence of their bacterial ancestry. It's like finding fragments of an ancient language embedded in a modern text, tracing back to where it all began.
And speaking of genomes, eukaryotic genomes are notoriously extravagant. They're often hundreds of times larger than prokaryotic genomes. For example, the human genome is about 700 times larger than E. coli's, but we only have about 4.5 times as many genes. So what's all that extra DNA doing if it's not coding for proteins? Is it just junk, as some once thought?
That's a great question, and it's a profound difference. For a long time, much of it was called junk DNA. But we now know that while some of it might be remnants of past genetic events or mobile elements, a significant portion is crucial regulatory DNA. This abundant regulatory DNA is key because it allows for highly sophisticated ways of controlling when and where a gene is brought into play. This is absolutely essential for complex multicellular organisms with diverse cell types. Think brain cells versus liver cells versus skin cells. They all have the same genes, but use vastly different subsets.
So that's about control complexity.
Exactly. Take the snapdragon, for instance, as mentioned in the source. A single gene mutation can cause leafy shoots to develop instead of flowers. Not because the flower-making gene itself is gone, but because the regulatory protein that controls when and where that gene is expressed has been changed, leading cells to adopt characteristics appropriate for a different location. It's about the exquisite control enabled by all that non-coding DNA, not just the raw number of protein-coding genes.
It's important to remember that many eukaryotes are still solitary cells, and they demonstrate remarkable complexity, not just multicellular ones.
You're absolutely right. We tend to focus on plants and animals, but single-celled eukaryotes like yeasts, amoebas, and paramecia are incredibly diverse and complex in their own right. They have incredibly diverse anatomies, from sensory bristles and cilia to stinging darts, and complex behaviors, like the protist didinium that can hunt and engulf prey almost as large as itself. In fact, single-cell eukaryotes are often more evolutionarily diverse than multicellular animals and plants combined. This highlights their long and varied history and their incredible adaptability, a diversity that we often overlook because they're not as visually familiar to us.
This brings us to a strategic choice biologists have made, focusing on model organisms. It sounds like a shortcut, but how does studying just a few species accelerate our understanding of all life, including ourselves? Are we risking oversimplifying human biology by relying so heavily on these model systems?
That's an excellent question to pose, and it's something scientists always consider. The idea isn't about shortcuts, but about strategy. Because all cells share fundamental properties, those universal features we talked about. The conserved genes studying a few accessible species, ones that are easy to grow, genetically manipulate, or observe, can unlock principles that apply much more broadly. It's not about oversimplifying human complexity, but about identifying core conserved mechanisms efficiently. It's like finding the universal engine schematics by studying a few well-designed car models before trying to understand every single custom car out there.
So let's start with the foundational model organism from molecular biology. What's the workhorse bacterium that started it all?
That would undoubtedly be Escherichia coli, or E. coli. It's a small, rod-shaped bacterium that normally lives harmlessly in our guts, but it's incredibly easy and cheap to grow in the lab, reproducing every 20 minutes or so under good conditions. Studies of E. coli and its viruses, the bacteriophages like T4 and Lambda, revealed fundamental mechanisms of DNA replication, transcription, and translation. These processes turned out to be remarkably conserved across all life, including humans. This tiny bacterium truly laid the groundwork for modern molecular biology.
And speaking of viruses, you mentioned bacteriophages. Viruses themselves are fascinating entities. They're not strictly alive, right? But they're everywhere, and they have a significant impact.
You're right, they're sort of on the edge of life, not strictly alive in the way a cell is. As they're obligate intracellular parasites, they depend entirely on host cells for reproduction, but they're ubiquitous and incredibly abundant, an estimated 10 to the power of 30 ocean viruses alone. Bacteriophages, which specifically infect bacteria, are particularly important, not just as research tools.
How so?
They serve as major vectors for horizontal gene transfer, which we discussed earlier. This is a huge contributor to rapid evolution in bacteria, including the spread of things like antibiotic resistance. A virus can pick up a gene from one host bacterium and ferry it into another, accelerating genetic exchange in a way that regular reproduction just can't match.
Okay, moving to eukaryotes. When it comes to the minimal model eukaryote, the choice is Saccharomyces, cerevisiae, common bakers, and brewer's yeast. Why yeast, and what does it tell us about ourselves?
Yeast is incredibly useful. First, genetically, it's surprisingly close to animals despite being a single-celled fungus. Second, it's easy and cheap to grow, much like E. coli. Third, it has a relatively small genome for a eukaryote, about 12.5 million base pairs, making it manageable to study. It's been invaluable for studying core eukaryotic-specific processes like the cell division cycle, how chromosomes segregate during mitosis and meiosis, DNA repair, fundamental stuff.
And the connection to us.
The amazing part is that human protein homologs genes with similar sequences that presumably evolved from a common ancestor can often function perfectly well when put into yeast cells. This means we can study the function of a human gene, maybe one involved in disease, in this simple, easy-to-manipulate organism.
And for plants, Arabidopsis thaliana is the model. What can a small weed tell us about plant biology?
Arabidopsis is a small weed in the mustard family, related to cabbage and radish. It's small, grows quickly, produces lots of seeds, and has a relatively small genome for a plant. It provides deep insights into plant development, how a seed becomes a plant hormones, responses to light, and plant immunity. It helps us understand how the fundamental logic of complex multicellular organisms evolved in the plant kingdom, which took a very different path from animals.
Now let's move on to the animal models. There are quite a few, each chosen for specific advantages. Can you walk us through them and why they're so significant?
Certainly. First, the nematode worm Caenorhabditis elegans, or C. elegans. It's tiny, transparent, and has clockwork precision in its development. Every single worm develops with exactly 959 body cells, and we know the precise lineage of every one.
Wow, that's specific.
That's specific. It's been crucial for understanding programmed cell death, or apoptosis, a fundamental process where cells deliberately self-destruct, which is vital for development and preventing cancer. C. elegans research also led to the discovery of RNA interference, or RNAi, a key mechanism for gene regulation found across eukaryotes. Then there's the fruit fly, Drosophila melanogaster, a true pioneer in genetics. Its study over a century ago provided definitive proof that genes are carried on chromosomes, thanks partly to its giant salivary gland chromosomes, which are easy to see. And it's been absolutely key to understanding how genetic instructions build the complex adult body from a fertilized egg, fundamental developmental biology. Even showing how a single mutation can lead to bizarre outcomes like legs growing where antennae should be, revealing the power of single genes to dramatically alter the body plan.
What about vertebrates? Flies and worms are quite different from us.
Good point. For early vertebrate embryonic development, the African clawed frog, Xenopus laevis, is invaluable. It produces large eggs that are fertilized and develop externally, making them easy to observe and manipulate. Cytoplasm extracts from its eggs are also powerful tools for studying fundamental processes like the cell division cycle in a test tube, outside of a living cell. Then there's the zebrafish, Danio rario. It's also great for genetic analysis, breeding quickly, and crucially, its embryos are transparent.
So you can see right inside.
Exactly. This allows real-time observation of developing tissues and cells, letting researchers literally watch organs form, cells migrate, and blood vessels develop. It's amazing for understanding how tissues are built and how defects might arise. And of course, the mouse, Musnosculus, is the predominant mammalian model. Mammals are genetically very similar. For example, the alpha chain of hemoglobin protein in humans and mice shares about 85% amino acid identity.
That close.
Very close. This allows us to use mice to model human biology and diseases. By creating targeted mutations in specific mouse genes, knocking them out, or altering them, we can reveal the function of those genes in a whole living organism, often mimicking human genetic conditions and providing immense insight into our own biology, physiology, and potential therapies. And finally, we shouldn't forget that we are a unique model organism ourselves, Homo sapiens. Obviously, we can't experiment on humans in the same way, but through self-reporting of genetic disorders over generations and now large-scale genome sequencing, with 8 billion individuals on the planet and any two differing at roughly 4 million DNA sites, we gain incredible insights.
How so?
We can correlate genetic variations with susceptibility to diseases, responses to drugs, and even trace human history, migration patterns, and ancestral interbreeding events, like with Neanderthals. It's a huge ongoing experiment in human genetics playing out across the globe.
And speaking of incredibly relevant and somewhat terrifying things, the modern impact of viruses, especially after the recent SARS-CoV-2 pandemic, really just can't be overstated. We've talked about them as tools and gene movers, but what about their evolution and the challenges they pose to us?
Viruses are truly ancient players. They're believed to have evolved from ancient parasitic RNA elements, perhaps even before proteins or DNA existed in their current forms. Their core functions are remarkably simple in principle. Replicate their genome and package it in a protective coat to get into the next host cell. But through billions of infectious cycles, constantly co-evolving with their hosts, they've developed incredibly complex and varied strategies to hijack cellular machinery and evade host defenses, making them formidable opponents.
In SARS-CoV-2 specifically.
Right. SARS-CoV-2, the virus causing COVID-19, has a relatively large RNA genome for a virus and produces about 29 proteins. These include structural ones that form the virus particle itself, non-structural ones that are part of the replication machinery, and crucially, accessory proteins that specifically interfere with our immune system's ability to detect and fight the virus. Understanding exactly which proteins do what and what makes this virus so much more dangerous in humans than many other related coronaviruses is an intense area of ongoing research. It's vital not just to combat this specific virus with vaccines and treatments, but also to prepare us for future threats from other emerging pathogens that will inevitably jump from animals to humans.
It really feels like by combining insights from all these models from humans and mice down to fish, flies, worms, yeast, plants, and bacteria, and linking them through the deep underlying commonalities of DNA sequence and cellular machinery, we're not just learning isolated facts, we're building a profoundly interconnected understanding of life itself.
Absolutely. It's the synergy between these different systems that's so powerful. A discovery made in yeast about cell division can shed light on cancer in humans. Understanding gene regulation in flies helps us decode human development. It all fits together because of that shared evolutionary history and conserved molecular toolkit. And the future challenge, which the source calls the great biosciences adventure of this century, is to move beyond these often qualitative descriptions to develop truly quantitative mathematical models of cellular processes.
Moving beyond diagrams to equations.
Exactly. Biological systems are incredibly complex, full of intricate feedback loops and interacting networks. To truly predict their behavior, how a cell will respond to a drug, how a mutation will affect an organism, we need robust numbers, precise measurements, and predictive equations, not just descriptive diagrams. This deeper quantitative understanding holds the key to unlocking even more of life's secrets, allowing us to not just describe, but to truly understand, predict, and maybe even engineer biological systems in predictable ways.
What an incredible journey we've taken together today. From the most basic, universal features that define all life, like DNA and protein synthesis, right through to the complex evolution of eukaryotic cells with all their internal compartments, and the profound power of using model organisms to unravel biology's deepest mysteries. We've seen how what seems so incredibly diverse on the surface is built upon the most astonishing constancy at its core.
It really is remarkable, and as you, our listeners, reflect on how deeply conserved this basic machinery of life is, maybe consider this. What fundamental principles might still be undiscovered? What completely new ways of organizing biological information or energy might be out there, perhaps in those unculturable microbes, or waiting to be revealed by new technologies in this push towards a deeper quantitative understanding of biology? And perhaps even more profoundly, what new possibilities does this ever-deepening understanding open for humanity? Not just in medicine, but maybe in engineering new biological solutions, or even in how we search for and perhaps recognize life beyond Earth. What are the next big questions?
Plenty to think about. Thank you for joining us on this deep dive into cells, genomes, and the diversity of life. Keep exploring the living world around you with curiosity, and we'll see you next time.